Hi, I'm George Hammond, conference producer at Technology Networks, and I'm here to introduce our next talk, Engineering Antibodies to Reprogram the Immune Response. I am pleased to welcome Dr. Jamie Spangler. Jamie is the Brodie Faculty Scholar and Associate Professor of Biomedical Engineering and Chemical and Biomolecular Engineering at Johns Hopkins University. Dr. Spangler earned a Bachelor of Science degree in Biomedical Engineering at Johns Hopkins University and went on to earn a PhD in Biological Engineering at MIT. Her lab, located in the Translational Tissue Engineering Center at the School of Medicine, applies structural and mechanistic insights to re-engineer existing proteins and design new proteins that therapeutically modulate the immune response. A very warm welcome to you, Jamie. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I will hand over to Jamie to begin her presentation.
Thank you so much, George. Appreciate the kind introduction and really the opportunity to showcase some of our research today. As George mentioned, I'm at Johns Hopkins University as an associate professor here, and I'm really pleased to tell you about some of our lab's work in engineering antibodies to reprogram the immune system. As many of you are probably familiar with, proteins have a lot of potential as therapeutics, and they've really been growing in the landscape since the early 1980s, when recombinant insulin became the first protein-based drug or biologic to be approved. And since then, the market has steadily grown to approximately 40% of the new molecular entities and new drugs coming out per year are actually in the protein or biologics category. And that's really exciting because whereas the market used to be completely small molecule based, we actually have a lot more biologics and drugs coming out in more recent years. And those drugs actually pack a punch, right? We have a lot of drugs in the biologics category, of course, antibody-based drugs shown here, as well as receptor decoy and other types of protein-based drugs that have made it to the top 10 drugs in terms of overall global sales. But when you think about proteins or antibodies or other sorts of natural molecules as drugs, there's actually nothing in nature, right, that's actually telling them to be good therapeutics. And in fact, there's a number of limitations of their potential as therapeutics. And the first limitation, of course, is that proteins in nature, right, are sort of programmed to be active on a variety of different cell types in a lot of different ways. And that can lead to difficulties in, the protein actually doing its given function, and also to harmful side effects from the protein going sort of off book. And then in addition, proteins can have expression or stability issues, including antibodies, which can lead to low recombinant yields, aggregation, degradation, things sticking together, falling apart, which can lead to downstream issues with developability, as well as immunogenicity. Finally, proteins, not necessarily antibodies, because they can overcome the very rapid clearance that happens due to due to their Fc domain interacting with Fc receptor, neonatal Fc receptor. The pharmacokinetics typically of proteins tends to be not great, and that limits their delivery efficiently to their target organ. And so really, right, as protein engineers, which my lab is very actively engaged in this field, our goal is to be able to redesign what nature has given us, or inspired by nature, actually design entirely new proteins that nature's never seen before that could be useful as therapeutics. And my lab uses sort of this three-pronged approach, the first prong being molecular engineering, which can mean a lot of different things to different people. What my lab does a lot of is what's called directed evolution, which is a procedure where essentially you take a protein shown in red here and make a bunch of different mutants or variants of it in a library, and then you use some sort of display system to present those different variants and then select them against a particular bait. In this case, it's this gray protein shown here. And then you pull out everything that works and you wash away the ones that don't. So through these iterative cycles of essentially Darwin in a test tube, you can actually make the protein do what you want it to do. And we integrate this with structural insights that come from either experimental methods, such as X-ray crystallography, cryo-EM, NMR, or with computational methods through collaborations that we have with various labs, either here at Hopkins or around the world, who take into account computational algorithms and use insights about protein structure from structure prediction algorithms, such as Rosetta, or through dynamic simulations like molecular dynamics. Various computational simulations that can be used to better understand those protein interfaces, so we can understand how to better design those libraries and ultimately get to a better answer for what our protein is. And then once we actually obtain those proteins, we can actually use structural insights, either, again, experimental or computational, to understand mechanistically how those are working so that we can make them better and really translate things from the discovery phase all the way through the design and development into people, right? To be able to translate those technologies, particularly antibody-based technologies, and then to loop back, say, this is how we did, and how can we actually move this forward and continually improve our workflow, with each iteration. And generally, my lab's research falls into sort of three different categories. The first is focused on engineering immune proteins, things like cytokines, growth factors, to modulate the immune response. The second focuses on designing these mechanism-driven antibodies that can serve as diagnostic tools as well as therapeutics. And then finally, building and constructing these platforms that not only our lab uses, but actually other labs around the world can actually use to advance their own discovery workflows and other sort of translational or basic research ventures through the use of these various platforms. So today's talk, I'm going to focus on a lot of our work involving antibodies, and I'll tell three different stories. The first is sort of in the immune engineered antibody category, where we engineered a cytokine and antibody to basically be a single chain molecule that's fused together and has particular biasing properties. We also did a second project that I'll talk about related to using antibodies as trafficking modulators, basically to cluster together receptors in the surface of the cell in order to affect the behavior of those receptors and ultimately of the receptor trafficking. And then finally, I'll talk about one of the platforms that we've developed, specifically what's called the biofloating platform for antibody discovery. Okay, so first things first, I introduced before that smaller proteins of the immune system, or smaller proteins in the human body, such as cytokines, can be very active in terms of dictating all aspects of the immune response, but they're limited by their size and their properties in terms of their pharmacokinetics. So cytokines signal through membrane-bound receptors. Typically, there's two of them, although there can, in some cases, be three. And it basically heterodimerizes or homodimerizes those two receptor subunits so that they come together. And when the extracellular domains come together, that causes the intracellular domains to encounter one another. And that leads to phosphorylation of the residues within the intracellular domain of those particular receptor proteins, which leads to a phosphorylation cascade downstream that ultimately converges on nuclear transcription of certain factors and genes that ultimately lead to responses such as differentiation, proliferation, migration, and survival. So ultimately dictating cell fate. And a particular cytokine that's going to be a major focus of the first part of this talk is what's known as interleukin-2 or IL-2, which was the first cytokine to be structurally and molecularly characterized, and for a good reason. It actually has a critical role in the development, growth, activation, and differentiation of a variety of immune cell subsets, particularly lymphocytes. And whereas IL-2 is, like other cytokines, activates receptors on cells through dimerization, its receptor actually comes in two different formats. There's a heterodimeric receptor, which consists of just the beta and gamma subunits. And then there's a heterotrimeric receptor that also includes this IL-2 or alpha subunit. And whereas the beta and gamma subunits are responsible for signaling, or the minimal active subunits, this alpha subunit actually increases the affinity of cells for IL-2. And so cells that express high levels of IL-2 or alpha have actually a 100-fold higher 10 picomolar affinity compared to those that express the heterodimeric receptor, which have only a 1 nanomolar affinity. And this is important because whereas effector T cells, things like CD4, CD8 positive T cells and NK cells actually express primarily the heterodimeric receptor prior to activation, regulatory T cells express constitutively high levels of IL-2 or alpha, so they primarily present the heterotrimeric receptor and signal in that way. Again, since IL-2 can actually act on both sides of the street, it's actually been used therapeutically for a variety of purposes. On the immune activating side, to activate effector cells, it's been used as a cancer therapeutic for treatment of infection. And that's actually been an approved drug since the early 1990s in metastatic melanoma and metastatic renal cell carcinoma. Unfortunately, very high doses are needed, right? Because the effector cells are much less sensitive and because the Tregs get activated by the same IL-2. And so it's sort of fallen out of favor as a primary treatment because these high doses lead to toxicities. So even though IL-2 as an immunotherapy can be very effective and lead to durable, robust cures in robust responses and cures even in 5 to 10% of patients, its devastating side effects such as vascular leak syndrome limit its applicability and its use in the clinic. On the flip side, people said, well, okay, if IL-2 at high doses is dangerous, what if we give low doses of IL-2 and bias it in that way towards regulatory T cells, which are more sensitive, and we could use that to treat autoimmune diseases, type 1 diabetes, Crohn's disease, colitis, psoriasis, MS, or for transplantation medicine, to promote tolerance towards a graft. And that's actually been used, low-dose IL-2, clinically for many years and is still undergoing active clinical study. But the limitation there, right, is everyone's low dose is going to be different. And that same IL-2 can actually stimulate effector cells, either if they upregulate the alpha subunit or if the IL-2 dose level is too high. So again, it's kind of this delicate balance. And ideally, we would want something that could actually bias IL-2 towards either an effector or regulatory response as a targeted therapeutic. And it turns out that there's actually molecules that bias IL-2, and we had to look no further than actually Western blotting antibodies. Owner Boyman in Jonathan Sprent's lab, when he was in Jonathan Sprent's lab, actually discovered that the ascorbyl-6 antibody, which is an anti-mouse IL-2 antibody, biases the cytokine towards effector cells. And conversely, the JES6-1 antibody biases IL-2, mouse IL-2 towards regulatory T cells. But of course, that begged the question of what was the molecular basis for this biased immune cell subset activation. So to this end, actually, when I was a postdoctoral fellow in Chris Garcia's lab, I had the privilege of solving the crystal structure of both ascorbyl-6 and JES6-1 bound to IL-2. And you can see here IL-2 flanked by its alpha, beta, and gamma receptors, respectively. And it turns out that ascorbyl-6 and JES6-1 are actually binding on opposite sides of the molecule and actually inducing opposite biases. So that was a very rewarding sort of thing to see. If you focus on the ascorbyl-6 mechanism, what the ascorbyl-6 is doing is actually blocking the alpha subunit from being able to bind. So it's also holding IL-2 simultaneously in a pose that makes it increases its affinity towards the beta gamma receptor. So it actually enhances the activity on IL-2 or alpha low effector cells. However, when it encounters an IL-2 or alpha high regulatory T cell, the IL-2 or alpha cannot bind to the IL-2. The ascorbyl-6 IL-2 can still interact with the same affinity with the regulatory T cells, but now it's lost its edge, right? Now that both sides are actually even on this playing field, you actually see that because the effector cells are more abundant, they actually get the overall advantage in terms of IL-2 availability. On the flip side, the JES6-1 antibody, which is biasing IL-2 towards regulatory T cells, is binding on the opposite side of IL-2, blocking the beta-gamma interaction. And the way that that molecule works is actually by blocking beta-gamma, it completely blocks the activity of IL-2 on the IL-2 or alpha low effector cells, so it's completely inert in that situation. However, on IL-2 or alpha high regulatory T cells, the IL-2 or alpha actually grabs the IL-2, displacing the JES6-1 antibody, which then diffuses away, allowing for the formation of the high affinity quaternary complex on the surface of the regulatory T cells with no activity on effector cells. That in turn induces an upregulation of IL-2 or alpha, leading to a positive feedback loop that ultimately exquisitely biases IL-2 exclusively towards regulatory T cells. So this is really great, right? That we understand how these molecules are working, and we've shown it has been shown that because of their biases towards either stimulating or suppressing the immune response, that in various preclinical models of either cancer or autoimmunity, S4B6 and JES6-1 are very effective, respectively. However, complexes fall apart, right? So just because we put IL-2 and S4B6 together, it doesn't mean they're going to stay together. And the dissociation leads to two problems. One is that dissociation of the antibody will actually decrease the half-life, because the Fc region of the antibody is what's sustaining the long circulation half-life for the cytokine. And how do we know what ratio? Right? If you actually give too much of the cytokine, then you have free IL-2 that was the same toxic problem in the first place. Or if you give too little, then you're not actually reaping the benefits of the bivalency and the presentation of the antibody, and that can complicate downstream therapeutic development. So I always say, if you can't beat them, join them. So that's exactly what we did. And we actually linked together the C terminus of IL-2 to the N terminus of the light chain of the either S4B6 or JES6-1 like immunocytokine or antibodies, and created an immunocytokine that is intramolecular, where essentially the IL-2 is binding within the antibody. So unlike typical immunocytokines, right, where the antibody is targeting towards some particular disease moiety, and the IL-2 or the cytokine sort of along for the ride on the back, we actually put it right on top so that it could interact directly with the antibody against that cytokine, and ultimately target that towards the particular cell type of interest. So again, to kind of see how we could translate this and move this forward, instead of using S4B6, as I mentioned on my previous slide, was against mouse IL-2, we actually replaced that with the 602 immunocytokine, which is against human IL-2. And we showed first that our molecule was actually intramolecularly assembling by showing a significant decrease in the binding towards free IL-2. We also showed that it completely blocked the interface and binding with IL-2 or alpha, but actually potentiated or left shifted the interaction with beta. Subsequently, we realized that our molecule, by replacing the S4B6 antibody with the 602, had actually lost some of its bias towards IL-2 or alpha expressing cells. So to this end, we actually used the yeast display platform to display the scFv of 602, and actually engineered that using air-prone PCR. We developed a library where we mutagenized the complementarity determining regions 1 and 3 of both the variable heavy and variable light chain, transformed those different variants into competent yeast, and then selected for IL-2 binding in the presence of competitor IL-2 or alpha. And what we hoped was that whereas the 602 scFv had some level of competition with IL-2 or alpha, we could actually shift that curve to the right, meaning that it would take more IL-2 or alpha to compete off the interaction between IL-2 and the scFv. So upon doing that, we actually derived an antibody from 602 known as the F10 immunocytokine. And this F10 antibody actually displayed the properties that we wanted in terms of increased bias towards effector T cells. And you can see here that particularly with CD8 positive effector T cells, you have a little bit of loss of activity, and you have some loss of activity on CD4 positive effector T cells. When it comes to Tregs, you're losing 5 to 6 orders of magnitude in terms of the response. So overall, right, your relative bias will be towards the effector T cells. And this plays out in vivo when you actually look at the expansion ratio of CD8 or NK cells relative to regulatory T cells. You can see that plain old IL-2 and even the 602 complex have very little effect on the ratio, and actually decrease that ratio compared to PBS, whereas you see a significant increase in the expansion of CD8s and NK cells relative to Tregs in the case of the F10 immunocytokine administration. So this was very exciting and promising therapeutically, but in addition to biasing the cytokine, we hoped to be able to target that towards the tumor microenvironment. So right around the time that we were thinking about this, Juni Shihara in Jeff Hubbell's lab led a paper where they showed that by conjugating and fusing IL-2 to a collagen binding domain, also known as CBD, the A2 domain of von Willebrand factor, they were actually able to achieve specific targeting of that molecule towards the tumor that ultimately led to improved anti-tumor activity of IL-2. However, building off of this work, we thought, can we actually make our F10 immunocytokine better by actually conjugating that to a collagen binding domain and then being able to target that towards the tumor. And so that's exactly what we did, making this fusion protein where the collagen binding domain was attached to the free C termini of both the the light and heavy chains. And ultimately, through intratumoral injection, we were able to very strongly retain the F10 immunocytokine within the tumor microenvironment. You can see that without the collagen binding domains shown in yellow, you have sort of gradual loss of that molecule and diffusion out of the tumor. Whereas with the F10 immunocytokine, although you have some loss due to metabolism, you severely reduce the inflection of that curve and ultimately end up sustaining the amounts of the F10 in the tumor. Even more importantly, we saw that there was absolutely nothing escaping, right? So again, while the amount of F10 immunocytokine would decrease over time in the tumor, we actually saw no escape of the F10 immunocytokine outside of the tumor, whereas the F10 immunocytokine showed a peak in 24 to 48 hours outside of the tumor. In addition, we wanted to see therapeutically what that would ultimately be able to do. And so we looked in both a B16F10 mouse model of melanoma, a syngeneic model, and also in a CT26 model of colorectal carcinoma. And in both cases, we saw significant reduction in the overall tumor growth, as well as very sustained or a sustained weight gain throughout the experiment. This contrasted with the F10 immunocytokine, which actually led to a drop in the weight due to the toxicity, again, because the molecule, even though injected intratumorally, was escaping systemically. So this was very exciting and really spurred us on to the development of potential combination therapies, looking at both B16F10 and CT26 with intratumoral injections of the immunocytokine combined with anti-PD-1 therapy, an immune checkpoint blockade inhibitor that's currently clinically approved in multiple formulations. And what we saw was that there was a strong synergy wherein when you have either the F10 immunocytokine shown in blue by itself or the immune checkpoint inhibitors by themselves shown here in the black dotted line, you get almost no improvement in survival in either of these models. However, if you actually inject the two together, shown with this red dotted line, you get significant synergy compared to either treatment alone, and ultimately significant extension of overall survival and even long-term survival in the CT26 case. So again, this was very exciting on the sort of immunostimulatory cytokine antibody fusion protein front, and we wanted to see if the same strategy could be applied on the other side to actually bias IL-2 towards regulatory T cells for immunosuppressive effects in autoimmune disorders. So to that end, we actually developed an immunocytokine known as the F511 immunocytokine, which fused IL-2 to a reported antibody co-developed by UCSF and Pfizer, known as F511, that actually has similar effects to JES6-1 in terms of that allosteric exchange, being able to bias IL-2 towards regulatory T cells. And again, we showed that it decreased the binding to free IL-2, indicating that it was properly intramolecularly assembled. We showed that the alpha subunit binding was pretty much unaffected by the F511 immunocytokine, whereas the binding to IL-2 or beta was completely ablated. That led to a bias in vitro looking at human PBMCs towards regulatory T cells. So whereas regulatory T cells were somewhat impaired in their activation by the F511 IC because it had to detach in order to allow for IL-2 exposure, we saw almost no activity on CD8 positive or CD4 positive T cells, which led to a strong bias in vivo for the F511 immunocytokine, this time in the opposite direction, biasing towards regulatory T cells over either CD8s or NK cells. And this ultimately was used in a mouse model of colitis to demonstrate that IL-2 treatment with the F511 immunocytokine led to a significant mitigation of weight loss, as well as decrease in disease activity index compared to either PBS, no treatment, or treatment with a control immunocytokine that just displayed IL-2 linked to an irrelevant antibody. And you can see that too with the colon histology. Again, the reduction in infiltration of immune cells and in pathology in the mice treated with the F511 compared to mice treated with the controls. We wanted to go even further and see if this could be used in a mouse model of type 1 diabetes. And we actually used a immune checkpoint inhibitor induced diabetes model to accelerate and standardize the development of disease in non-mice. And what we saw again was that the anti-PD-1 plus the F511 immunocytokine treatment shown in purple compared to just anti-PD-1 by itself with PBS shown in red, or anti-PD-1 plus the control immunocytokine shown in black, led to a significant slowing and delay in the onset of diabetes, and ultimately led to long-term resistance to diabetes development, here showing 6 weeks of treatment. And we went even further, actually out to even 6 months of treatment. And we saw that actually over 50% of the mice who had received the F511 immunocytokine were resistant to development of diabetes, not only from induced disease from treatment with anti-PD-1, but even compared to mice that were not treated with anti-PD-1 and actually had spontaneous disease. So it was protecting against both spontaneous and induced disease. And we were very excited to see that result and have been working on the clinical development of that molecule in addition to the F10 immunocytokine as potential therapeutics. So the next story, and I'll touch on this and the third one pretty quickly for the sake of time, but happy to elaborate more in the Q&A. Regards a recent story that we have on designing multiparatopic antibodies that regulate molecular trafficking. And this was based on the premise that designing a multi-specific antibody that targets multiple epitopes on the same molecule can actually affect the trafficking behavior of that molecule. So the concept is quite straightforward, and that is that an antibody, because it's bivalent, can cross-link together 2 receptors on the surface, but that's it, right? That's endgame, because both of the epitopes are taken. However, if you come in with a second antibody that binds to the same receptor at a different position, you can actually extend that daisy chain, right, to get these sort of long clusters, theoretically, of infinite length all over the surface of the protein. And this is important because this can actually affect aspects of the molecular trafficking of the target protein. In this case, specifically, it can affect endocytosis, causing a molecule to actually go in to the cell much faster. This was demonstrated in the case of epidermal growth factor receptor back in my PhD work. And it can also knock out or ablate receptor recycling, basically shunting everything towards the degradation pathway. And this can actually be achieved either by a combination of antibodies, right, which are against different epitopes on the same receptor, or by creating these multiparatopic antibodies, as they're known, that have multiple different specificities against a single receptor at different sites or different epitopes. And so ultimately, we developed these multiparatopic antibodies that have essentially one arm that's ligand competitive and the second arm that's actually non-competitive with the other arm, and leads to ultimately downregulation of both the ligand and the receptor. And what we did in this work was we actually chose to apply this to the sort of immune checkpoint inhibition concept, that you could actually use these against immune checkpoint inhibitor proteins to establish a more complete blockade. And what I mean by this is, right, normally T cells get activated by antigen-presenting cells through 3 signals, signal 1 being the central complex between the TCR and MHC, signal 2 being B7 or other B7 family ligands interacting with CD28 or other co-stimulatory molecules on the T cell. And ultimately, that leads to secretion of various cytokines that will ultimately allow for the T cell to undergo sustained expansion and activation. Unfortunately, that same activation that happens leads T cells to upregulate a variety of immune checkpoint proteins, in particular PD-1, which can then in turn be activated by tumor cell presented PD-L1 to lead to T cell exhaustion. And so a promising treatment that's been developed, of course, is these monoclonal antibodies that are clinically approved, as was mentioned before with immune checkpoint inhibitors, against either PD-1 or PD-L1 that block the interaction, right, directly between those 2 molecules. And then what we thought was, can we actually take this a step further by completely eliminating PD-1 from the surface, right, sort of mowing the lawn through these multiparatopic antibodies that would actually downregulate and manipulate the trafficking of those receptors. And so whereas PD-L1 interacts with atezolizumab, shown in red here, which is a clinically approved antibody that blocks the interaction between PD-1 and PD-L1 by binding to the interface, right, and blocking and sterically occluding the binding of PD-1. We wondered if we could discover molecules that would bind somewhere else and could be used for trafficking downregulation. Or trafficking mediated modulation and ultimately downregulation. So we selected two libraries, an scFv library developed by Ryan Kelly in Dean Wittrop's lab that is designed to have less nonspecific binding. That's called D12. And then we also used shark immunization. So we collaborated with Helen Dooley, who is at the University of Maryland, and we're able to inject nurse sharks with our target, in this case PD-L1, and derive two distinct nanobodies of nanomolar affinity that actually bound to different epitopes on the PD-L1 receptor, and so, or on the PD-L1 ligand. And so ultimately, right, our goal was to develop things that would bind in other places relative to atezolizumab. It turned out that the D12 scFv that we first developed bound in the same spot and was competitive, so that was not helpful to us. However, we actually discovered these two new VNARs, which are shark antibodies that are single domain, similar to camelids. Sharks have single domain antibodies. And these actually bound on the side, right? They kind of got into these like nooks and crannies on the sides of PD-L1, and were actually non-competitive with both one another and with atezolizumab. We went crazy in terms of developing all of the different types of biparatopic antibodies that could come from these four different anti-PD-1 or PD-L1 antibody fragments that we developed. And ultimately, we found that our winner was actually a molecule known as VS15 that conjugated the the C terminus of the VNAR B8 to the N terminus of the light chain, or sorry, of the heavy chain of atezolizumab. We then went a step further and actually said, well, we have three different specificities, so why not try making a triparatopic antibody, which is exactly what we did. And we developed all these different triparatopic antibodies targeting the PD-L1 at different locations. And ultimately, the winner amongst all of our triparatopic antibodies was this molecule called TS1521, which has, again, based off of the other but winning biparatopic, which has B8 at the N terminus of the heavy chain. We also now have A9 at the C terminus of the light chain. And long story short, to keep on time, we compared the TS1521 triparatopic antibody to atezolizumab, and we saw a very distinct pattern of PD-L1 clustering, which you can see here under confocal microscopy with these green clusters sort of all over and throughout the cells. We also showed that this led to increased co-localization with both endosomes and lysosomes for TS1521 relative to atezolizumab. And you can actually see in a time-lapse microscopy the development of these clusters over time as you add in the TS1521, again, throughout all planes of the cell. You can see those receptors rapidly clustering and getting internalized. Ultimately, we wanted to see what this would do in terms of being able to block the PD-L1 on tumor cells more effectively. And so to this end, we actually performed these radiolabeled peptide experiments where essentially we used a radiolabeled peptide that binds to PD-L1 in the epitope that would normally bind to PD-1 to basically quantify the amount of bioavailable PD-L1 on tumor cells. And the idea was that when we introduced our TS1521, our multiparatopic, we would have fewer sites that were accessible to the radiolabeled peptide. We then performed an experiment where we injected mice containing an orthotopic breast cancer tumor with either the radiolabeled peptide, or sorry, with either TS1521 or with saline. And again, you can see that TS1521 led to significant reductions in the amount of bioavailable PD-L1, and that even after 96 hours, even though there was a slight rebound, there was still a very significant and dramatic drop in the levels of PD-L1 that were available for signaling. Finally, we performed an activation study to make sure that our T cells could actually get reactivated by this treatment. And to this end, we looked at PBMCs from patients with hepatitis C and who were also CMV positive. We stimulated those cells with CMV peptides and showed that these patients who had upregulated levels of PD-L1 upon treatment with our TS1521 compared to atezolizumab led to significantly more interferon gamma secretion reflective of increased T cell activation. Okay, so in the final few minutes, I want to cover this third area, which is the development of a discovery platform against membrane proteins. And to this end, just to motivate the problem here, membrane proteins constitute only 23% of less than a quarter of all proteins in the human genome. And yet they actually make up for 60% of targeted proteins in therapeutics that have been developed as drug targets. And the reason that they sort of have this outsized significance for therapeutic development is, of course, because they're transmitting information from outside the cell to inside the cell. And so they play a very important role in a variety of different processing and physiological mechanisms. Antibodies, of course, need no introduction as their importance in the pharmaceutical industry and their capability of binding to precise sites on their target antigen, being able to affect multiple mechanisms, and having a long sustained half-life thanks to FcRn-mediated recycling. However, the discovery of antibodies against membrane proteins has been very difficult, and the main reason for that is that the methods that we're using to discover either immunization, as we did to the sharks, or display platforms, as was mentioned before, such as yeast display and phage display, all require a soluble protein as the selection target, right, shown in gray here. And that can be very difficult for a membrane protein to actually purify that recombinantly. And even when you do, it's actually in its non-native orientation, and you can get things binding to the inside or outside, which is not, intracellular binders would not necessarily be relevant in the case of an antibody. Finally, post-translational modifications can be different depending on how these things are actually expressed in their native conditions versus when you're actually presenting them in the context of recombinant protein. So our lab decided that it would be actually very valuable to actually cut out the middleman, and instead of purifying the protein from cells, we could actually just present the cells themselves and actually discover antibodies or single-chain variable fragments thereof that could actually bind to this protein on the cell, right, in real life, in the way that it would be physiologically represented in the organism. And so we hoped to be able to merge together these target displaying cells with the antibody displaying yeast to create this high throughput platform for antibody discovery. Now, we're not by any means the first people to do anything like this, but what our contribution was whereas in the past, people used this method called biopanning, where they basically just grew a monolayer of cells through a bunch of yeast displaying different antibodies on top of them and said, okay, what binds? What we wanted to do was actually do everything in suspension. And the idea was that we could get more dynamic range by having everything in motion and get more sensitivity for binding. Also, another ulterior motive that we had was it would actually be much easier to quantify things because you could quantify everything by a mechanism like flow cytometry instead of trying to count, right, all the individual yeast binding in these different situations. And so, again, we wanted to look at interactions that would be happening at high affinity, medium affinity, low affinity, or for a control where there should be no control scFv, where there should be no binding. You can see that, again, with biopanning, you can kind of see this visually through microscopy, but biofloating allows you the advantage to actually cut out all of that time analyzing by microscopy and actually just analyze and quantify much more precisely using flow cytometry. Again, the increase in sensitivity of the system was showcased both when we titrated based on affinity or on avidity. looking at either dense presentation on the mammalian cells or sparse presentation on the mammalian cells versus high versus low affinity scFVs on the yeast side. And on both sides, right, the yeast side and the mammalian cell side, we saw that there was much more sensitivity in the biofloating context, that it could detect dense, intermediate, and sparse, whereas the biopanning was much worse at even detecting the dense at all, and could barely detect anything for sparse and nothing for intermediate. Ultimately, we wanted to use this to create a discovery platform. So we'd be able to select from a naive library clones that were actually binding to our mammalian cells using just generic flow cytometry and magnetic activated cell sorting based technologies. So to that end, we actually developed a workflow that involved a first step of basically clearing away anything that would not be binding to our target expressing cell, to things that would be binding to our target null cells, and then selecting for things that were specifically binding to our target expressing cells, not to the cells themselves, but rather to the specific target on our cells. So to that end, we actually labeled our target null cells in a first step to pull out anything that would bind there as non-specific binders. And then in a second step, where we had target null cells as competitors, we added in fluorescently labeled target expressing cells, which could then be detected and sorted out through either magnetic or fluorescence activated cell sorting, discarding the specific, non-specific, and non-binders, and ultimately being able to regrow our specific binders. And we utilize this workflow in several contexts against various GPCRs. This is looking at CXCR2. We found a variety of clones that were binding to target expressing cells, but not target null cells, using a particular metric of yeast cells per complex, reflecting the extent of enrichment over the course of various rounds of selection. And you can see that ultimately we got about 27% specific binders, including a molecule that actually bound to CXCR2, known as CX2, at a unique site compared to the 10H2, and showed lack of competition with that molecule. So that was very exciting to find this new binder to the CXCR2 IL-8 receptor. We then got greedy and said, can we use this in general, more generally against GPCRs? And indeed, we found against 3 different GPCRs shown here that we got high numbers of specific binders and actually very few non-specific binders in all 3 cases. And some non-binders, of course, things that didn't bind anything at all. But generally, what we saw was what we got, which was very exciting, with an overall on yeast specific binder rate of 72%, and then 80% of those that we found on yeast actually bore out when we looked at recombinant proteins on the cells themselves, looking at the antibodies. So this was very exciting, having very low attrition rates, and really what you see is what you get in terms of the binding to mammalian cells. Okay, so to wrap things up, I've talked about 3 stories today: these cytokine antibody fusion proteins for biasing IL-2, these multiparatopic antibodies against PD-L1 that can be used as immunotherapeutics, and then finally this new platform for antibody discovery against membrane proteins from naive libraries. So I'd like to end by thanking the amazing people who actually did all of this work, certainly not me, and certainly much better than I could have ever imagined doing it myself. Thank you for your attention, and happy to answer any questions.
Thank you so much, Jamie, for that incredibly fascinating talk. We really do appreciate you sharing your expertise with us today. As with all the talks, we're likely going to have lots of questions come in from the audience, and I can see that we have received some already, so I'll move straight onto the Q&A. I am aware of the time, so we unfortunately only have time for 1 question today. However, any questions unanswered will be addressed offline. So Jamie, your question is, what are the greatest challenges that you face in clinically translating therapies?
Ooh, great question. So a major challenge, in terms of translating the molecules is really the models that we have to sort of accurately recapitulate how a molecule is going to perform in the clinic. So one major limitation is that the mouse system, right, which is usually used in preclinical work, is very different from the human system, to the point where, especially with creating molecular drugs, there's not cross-reactivity between the human and the mouse versions of the proteins. And so when you actually do this mouse studies, sometimes you have to create a surrogate molecule, which won't necessarily reflect what the real molecule will do. Or even if you're using the actual same molecule and you have some cross-reactivity, the cross-reactivity won't be exactly the same, right, as it would be in the context of a human. And so it's very difficult to gauge, right, how it would actually perform in another species. So I think that there's a lot of different microphysiological systems that people are developing, organoid models, different ways that people are trying to overcome those problems, but really trying to recapitulate the sort of complex environment, particularly of the immune system, in these preclinical models will be critical to developing the next generation of drugs.
That's Unfortunately, that is all that we have time for today. Just a reminder to the audience that any questions we didn't have time to get to will be sent to the speaker after the event. As with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thank you once again, Jamie, and we'll be back soon with the next talk.
Thank you so much.